So up first, we have got Tao Lu, who's going to talk to us about Max. Take it away.
Thank you. So hello everyone. So thanks for having me to present our project. And now I want to introduce the Max 3 project that we are currently building under the support from CZI. Our aim is to build a flexible and versatile big collar for the gene regulation studies. As I mentioned in the pitching period, so actually Max is a very domain specific tool. So when we published the paper in 2008, about already 12 years ago, that time I was a postdoc working in Dana-Farber Cancer Institute with Shirley Liu's lab, and at that time we get almost the first bunch of ChIP-seq data, and we want to develop a tool that's specifically designed to analyze only ChIP-seq data alone. But after we published this on genome biology, and over the years, and I saw a lot of people that try to use Max to analyze all kinds of different data, including like taxic and histone ChIP-seq data, which we do not originally design for. So at that time, it's mainly developed and maintained by the team. But after I left this Max lab in 2013, I started my own faculty position at Buffalo, and the fund to Max is discontinued in 2014. And basically, the tool is mainly maintained by myself in my spare time, as I mentioned. So, but we are so surprised by looking at citation over the year, even after I left the lab in 2013. So you can see the citation for Max is just increasingly continuously, and now it just hit 8,000 citations, and every year this year we also get like 1,500 citations. So the thing for Max is that it's a very straightforward and simple tool. It's just to do peak calling on the so-called functional genomics data. And it has been, because I was working in the ENCODE consortium, so the tool has been incorporated also in the ENCODE pipeline to analyze the ChIP-seq and ATAC-seq data. And also is already included in also other platform, also supported by CZI, for example, Galaxy platform. So we are so surprised to look at this. So we just, over the year, even without any support, I keep getting a lot of like questions, suggestions, and ideas from the community, which just make me feel like this open community actually work to just push this software get better and better. And I got a lot of feedback and implement the bug fix and implement the features still using my spare time because I don't have any funds to support this work. And when I see CZI grant, I just try to apply for it because it will make me just more comfortable to just devote my time to make this better. Because over the years, I saw a lot of suggestions and questions from the user community. There's a huge user community out there, and I still keep receiving a lot of feedbacks every day. So let me first talk about the basic idea behind Max. Actually, it's very straightforward. At the very beginning, when we designed this kind of tool for analyzing the ChIP-seq and ATAC-seq data. So there's basically two models using Max. If you consider about the data, one model is to just try to smooth the data. Another model is try to normalize the data. So the way we smooth data is that we only consider, we talk closely to the biologist. So the success, I think, for Max is that we keep very good relationship with biologists and keep hearing from them and what they want and what they think that the data should works like and what kind of systematic bias they thought in the system. So the first model on the left is basically if you look at the problem we want to solve is that well on the genome that is highly functional or actually bound by certain kind of protein to have like gene regulation functions. So whenever this position is located, you should imagine that biologically there must be a protein attached to the DNA or just close to the DNA, and that part of the DNA is protected from outside. So when you do an experiment, for example, to share the DNA, that particular region is protected, and what signal you get is surrounded. So the first model we use is to just make the model of the signal around this binding site to smooth the data, so that the enrichment of the signal right in the middle of the potential binding site. The second model we used is to normalize data that we consider about systematic bias or noises in the system. For ChIP-seq, we estimate this kind of systematic bias from input and control data, which we do not have any kind of manipulation on it. For example, genomic input, just get the DNA and the sequence out without any kind of experimental thing to just cross-link the protein to the DNA or fish out the protein. And that can give us a very good background of it. So we use a so-called local bias. We summarize from the surrounding region in each genomic loci, and use a so-called local Poisson test to calculate p-value and assign a score for each base pair. At the very beginning, when we published the first version of Max, we cannot do this in base pair resolution. And right after, I think 2014, and I just make this Max version 2 to just try to calculate the score for every single locations, which actually just increased this kind of popularity of Max, because now people just think, just by using this kind of very simple strategy, you can just deal with all kinds of like signal enrichment question in the genome and try to call the so-called peak while the signal is highly enriched compared to the background. So over the years, I saw a lot of users just feedback to me and ask me, can I use the Max tool to analyze other type of data? It's just making me so surprised, because originally, as I said, we particularly designed this Max tool to analyze the so-called non-histone ChIP-seq. And then we just keep adding more and more functions to analyze the so-called histone ChIP-seq. And when people just jump to the so-called ATAC-seq kind of technology, which require less material to do the experiment, and is getting more and more popular nowadays, because you can easily just get like hundreds or thousands of cells from the patient and just get the experiment done and get all the possible potential functional elements in the genome. And also, if you're working in single cell experiments, single cell ATAC-seq is also very mature technology right now. It's commercialized, and people can just using like $2,000 and to get their own single cell ATAC-seq data on their own the biological sample they are interested in. But I also see a lot of feedback from users. They want to use Max to analyze, for example, cut and run. And also, to my surprise, is that some people asking me, can I just use this tool to analyze RNA-seq? Because this is just totally different kind of assay. They thought that's after you get all the sequencing reads and map back to the genome and you get a signal track, you just call the peak, and you will know where the gene, where on the genome is transcribed actually. So I just got a lot of user cases from the user community, but I keep telling them, for example, RNA-seq, you should have a dedicated tool to just analyze the data. Don't just try not to use Max. But they also give me some kind of impression is that we have a lot of things to do, not only like dedicated RNA-seq tool that I'm not taking responsible of that, but also for Max, if you want to use our tool to analyze different kind of data, we should think about different set of parameter or just tweak the algorithm to fit that kind of data analysis, which originally I don't have time to do. So that's why at a certain stage of the development, I split the Max tool into totally modularized kind of structure. I divide the Max tool into like 13 different kind of modules. Actually, the current module is in the inner cycle, and I currently, for the Max 3 project, I will give an update later on, is that we add another module called call variation module. And And all this module can work together, for example, to normalize data and to call the peak specifically from certain type of data. And user can just flexibly just combine all this tool to make their own customized workflow. For example, in this case, people can just get the data and use the filter duplication module and get a result and predict the fragment size and do the pi up track and compile the signal to the noise and just run the peak call, and to just implement the same function as in our main function we call the call peak function. So we designed this to make it more flexible. And there's another update after I try to apply the CZI grant in the cycle 1, because in the cycle 1 I also applied it, but I didn't get it, and I got it in cycle 2, is that I start to just try to make Max work in implement the CI/CD for Max, because for most of the tools in biology, bioinformatics field, the lack of maintenance and lack of robustness of the software, sometimes just student update it and it just breaks the whole code. And for Max, since we have a huge user community, I want to make this tool very robust. So right when I apply for the cycle 1, I start to build the CI/CD using Travis. But now I just switched to GitHub because I don't want to buy the credits in Travis, and GitHub Action just give us like a lot of runtime on that. So currently I implement Max can be successfully tested and run on all kinds of different kind of CPU architecture, including x63, 64, and also ARM architects, and also other kind of CPU architects, with the help from June and Michael, who are also supported by CZI, working on NF Core and CWL. I got a lot of help already from the community already. And another update from the current Max 3 project is that currently we moved Mac. We built a package for Max in R. Actually, in our department, my neighbor is Martin Morgan, who led the bioconductor core team. He asked for this for years. So finally, I got the capacity and also developer to implement this Max R port. And currently, the R package is packaged up, and we prepared the testing data and also submitted already to bioconductor. Currently is under review. Another update from Max 3 is that currently I add a new module in Max, which is also based on the idea of calling peaks. Over the years of our experience on the data analysis is that we saw a lot of enrichment of the sequencing reads at the so-called peak that called by Max. Sometimes the peak region, as I showed on the left side, has like hundreds of reads pile up together. So that means if you use only the sequencing reads you get from a sequencing machine without any help from the whole genome sequencing, and also you do not need to just check the load the reference genome sequence in the memory, you can already call the variation directly from the reads from the chipseq. So we built this module, we call it call variation module. We get the sequencing reads at the peak region, and we use Henley's tool called Fermilite to assemble the whole fragment and compare it to the reference genome sequence we can also get from the bound file, and to call the variation and apply to this model and predict where the variance is. So the prediction power is very good if you compare to GATK. So basically, in the peak region, we can reach a F2 score almost larger than 0.95 if you're dealing with a high coverage chipseq data set. On the right side, you can also see that we can also get very, very promising biological insight from this type of analysis. On the top is a so-called CTCF protein DNA binding site, which has a very strong binding pattern at this 4 and 5th and 9, 10, and 13, and 14 position of this binding location. And from our analysis, we directly get this kind of insight because of this position. From our analysis, we can find that if this position has any kind of variant called from our algorithm, then they will totally change the so-called allele-specific binding, which give a binding in one of the chromosome, but another not in another chromosome. Because if you think about human genome, we have double copy of the chromosomes. So previously we implement this tool in a codename called Sepper, and now we just merge this into Max. And then let me talk about some future developments. Sorry, I think it's already 17 minutes. So we'll add a dedicated tool module for analyzing taxic data, and also add a dedicated tool for differential peak calling. I'm a fan of DSeq too. I saw Michael Love is here. So yeah. So originally our plan is to just make a link from our Max to DSeq, but I don't think this is a good way to just reinvent the wheel. So I'm thinking different kind of strategy. Maybe I will come up with this idea and discuss with you guys later. And the next module is to build the matrix module, is to prepare the single cell taxic data right for the single cell analysis. First, let me talk a little bit about the hidden mark model, the ataxic analysis. This is a published paper last year, made by my student, who proudly announced himself as the first Mohawk bioinformatician. And he made this using the hidden mark model to better analyze the ataxic data, which is the only dedicated tool nowadays for analyze the ataxic data. So I will skip out the detail of how they implement that. And another progress for Max is that we just recently published a paper together with Shirley Liu Lab and Dana-Farber, which is called Maestro. We build an integrative analysis of single cell assays, including the single cell RNA-seq and the single cell ataxic, and we tune and incorporate Max also in this single cell ataxic analysis pipeline. So we'll just keep improving both Max and Maestro, let them work together, because everybody are a fan of single cell analysis right now. So I will just put my focus more on this direction. So at last, I want to thank the support from CZI, EOSS. Without that, I just do this work all by myself in my spare time. And now I have some support. And I want to thank my core team members, Chang who made this report, and Jie Wang who made the documentation, and my collaborators at Shirley Liu Lab. And most importantly, I want to thank all the Max user community who give me the feedback over the 12 years. Thank you.
I guess I'm curious what you're thinking about differential peaks.
Yeah, I think, whenever people ask me, I say you should just get the count from the peak and just use DSeq too. Simple enough. But still, I'm thinking that for ChIP-seq and ataxic analysis, we should also think about the shape change within the peak, the signal shape change, which cannot be solved in current RNA-seq differential RNA-seq way. So I'm thinking how to evaluate the difference between the shape of the enrichment shape within the peak, and come up with a new way to call the differential peak calling. So we are working on that part. Thanks for the question.
I have a question. What kind of coverage do you need to get robust variant calling?
So currently, I think for about 100 reads are pinned together in the single location. But let me tell you that even if you have like very common ChIP-seq assay right now, you should have enough coverage at the peaks. Because at the peak region, all the regions enriched there, you already get huge enrichment compared to the whole genome sequencing. So there's already enough information out there.
This is Aaron Quinlan, just a follow-up question about variant calling. You may have mentioned it, but I probably missed it. Have you compared the variant calling that you get out of that pipeline to some of the other approaches that you might use, just sort of off-the-shelf variant callers that look at any coverage greater
we compare with JTK. It's performed similarly, I should say. But yeah.
Yeah. Okay. Thank you. I missed that.
Thank you, Aaron.
All right. With that, thank you very much, Tao. And up next, we've got Kimberly Pagel. Take it away.
So a brief reminder from the pitch. OpenCRAVAT is a variant annotation and analysis tool. Our goal is to make variant analysis easier and more customizable with open and modular software. But to get really started and learn more, let's get right into the demo. I'm working with Dr. Gottlieb Sen at Johns Hopkins University, who is well-versed in congenital heart disorders, and she's got this great data set of 352 patients, whole exome sequencing of them, who have congenital heart disorder. So that's the data we're working with, and our goal is to find disease-related variants among these patients. But since we don't really have any control folks to compare to, but we do have subdivisions of the phenotype, we're going to try to find disease-related variants in the subset of patients who have coarctation or narrowing of the aorta. So let's see if that's doable with OpenCRAVAT. There are going to be 4 steps in this analysis pipeline. First, since it's a modular architecture, we'll need to select what annotations to use, what's relevant to the analysis. Then annotate the variants and put these annotations right next to the variants. Third, filter the data. And then fourth, analyze and find relevant disease-causing potentially variants. So the first step is selecting annotations. If we step back and think objectively about this, what kind of information would be useful if we've got whole exome sequencing of 300 people? Well, it would be rare variants that would be more likely to cause the phenotype, because if they're common, everybody would have congenital heart disease. It would also be useful to narrow down to variants within CHD-related genes, or genes that are transcribed in the developing heart. If they're not transcribed in that region, they're not really interesting to get into. So how can we do that? Let's take a look at the store. So this is what the graphical interface looks like, and I'll be toggling back and forth between the interface and the slides. To get to the store, click on the Store tab, and you'll see all the resources that are available, broadly classified into reporters for different output formats, annotators for information, and visualization widgets to see what's going on. So let's look at 4 different kinds of modules here. The first, we want to know what variants are rare, and I happen to know about this resource called Nomad. To go back a step, this store is very large, more than 150 different components in here. So these tags along the left-hand side categorize available resources into little groups that are more helpful when someone's trying to explore. So for example, allele frequency includes things like the 1000 Genomes Project, some population-specific databases, Nomad, the newer release of Nomad, and some older data sets as well. I'd like to go with Nomad here because it is pretty well represented with exomes compared to Nomad 3, which has better coverage of genomes. So if we go down to the cardiovascular tag, which we've been able to propagate with our work with CZI, there are a few options here, but one of them we developed particularly for this project called Index Congenital Heart Disease. So this includes several networks particular to the development of congenital heart disease and the way that these partners interact in the developing heart. All right, we'll include that. Then it would be great to know if any of the variants are already known to be related to disease, since there's a large corpus of work for that. So if we go to the clinical relevance tag, there's quite a few things here, but I know offhand that these 2 are great. ClinGen Gene is a very well curated set of genes that are known to be related to diseases. And ClinVar, of course, is a database of variants that are known or likely to cause disease and which disease. And finally, one more thing to squeeze in is non-coding. There was this amazing tool developed called Segway, which tries to be an encyclopedia of human regulatory elements. It can find regions that are transcribed, that are in heterochromatin or quiescent, not expressed at all. So including that will give us a little bit more of a sneak peek into which variants are more likely to be relevant in our exome sequencing data. So to recap, we've got 5 things: Nomad, Index, ClinVar, ClinGen, Segway. We could include a lot more, but that would be overwhelming for the purposes of this demo, so let's keep it simple. we could easily include every single thing available. That wouldn't be too computationally advanced for my laptop, but it would be too much for me to handle. So let's not do that. We're going to include one more annotator, and it's something that we've developed since starting working with CZI called case control analysis. Frequently, analyses will involve many patients, and those patients can be subdivided into groups. Sometimes the cases are phenotypically neutral or just don't have the phenotype of interest, but other times we've got data sets of patients with subphenotypes. And it might be interesting to try to tease out the differences on the genetic level. Say there are certain genes that are more mutated in one group than another, or certain variants that tend to pop up much more frequently in cases. So the case control takes an input file, as shown on the right, that has identifiers in the left and a column on the right that labels each thing as case or control. P-values are assigned with a Fisher's exact test using a contingency table for dominant, recessive, and allelic inheritance. Trying to get a broad swath here so that we can help find variants that are differently presenting in cases versus controls. The next step is to annotate the data. I personally run it on the command line. It's easier. It's the world that I live in most of the time. And here's how that command would look on the command line. OC run, OpenCRAVAT is OC, run, file name, -L, and then the genome version. Very important to get that right. -A, and then all those annotators that I had mentioned, ClinVar, ClinGen, Nomad, case control, segue, and index. And then since we're doing something really special here with the case control, we say we want to do the case control analysis and include the case control IDs file. But let me show you how it would work in the interface so I can prove that this is really biologist friendly or suitable for teaching. It has a very similar setup. Use this dropdown to select the genome version, click a button to load your file, or click an example file if you're running a demo. Looks like this. The annotations list is propagated from whatever I've got it downloaded on my local computer, but this also can be run in a web server at run.opencravat.org. So here I've selected some random things just because I have so many things downloaded, but here's index congenital heart. And the final step is to upload the case control file. So click that button and add the file. But for the purposes of this example, I'll keep it simple. Click the big orange red annotate button and you'll see that it's added on the table with the status of starting. As the file is processed, it will update. See, it started converting the VCF into an internal usable file format. Now it's mapping the variants from 19 onto 38, and it's done because that was an incredibly small job that only has 4 variants and 3 annotators. For the file that I ran, it took a little bit more. It had almost 1 million variants, so it took an hour or two, and I ran it with those 7 annotators. Now that the annotation is done, we have the button here that says Open Result Viewer that lets us pull up the results and analyze them in the interactive browser. We could also download the output files in VCF, TSV, CSV outputs if you'd like to use it in a pipeline. But since this is a demo and command line stuff isn't as exciting as an interactive GUI, let's take that route. The next step is to filter the data. We've got 1 million variants, and that is too many to look at manually. So I created a filter to remove some of the stuff that's less interesting, at least on a first pass. So this looks complicated, but it doesn't have to be complicated. There's a very simple interface over in this little button that can quickly cut down allele frequency, only grab variants of particular types. It's not really a big deal. But the query builder allows for really powerful filters. So let me just explain what this is and make it seem not so bad. This group down here is saying if ClinVar knows that it's benign, don't include it in our analysis. This group is saying make sure that someone in the case, either heterozygous or homozygous, like at least 2 people have it. That way, it's not some one-off random variant that might not be related, and to cut the numbers down for the purposes of this demo. Then we went to Nomad and either took values that were empty, which means they don't have annotations in Nomad, so they're pretty rare, or an allele frequency of 0.001, so they're quite rare. And finally, I did some magic to take out non-coding variants and synonymous variants, again, for the purposes of the demo to keep it clean. So with only those like 4 things, I've taken it down from 1 million variants to 2,500-ish variants, much more tractable for analysis. And I haven't even really gotten into any of the cardiovascular specific annotations. So enough talking about the interface. Let's look at these variants. These people were so nice to give the data. So there's 3 tabs at the top, summary, variant, gene, that give you different lenses of thinking about the data. The summary tab is a high-level view of what has been seen. So this changes to reflect the filter. We've got a sequence ontology plot that shows that, unsurprisingly, the majority of the variants are missense, and then deletions and insertions are the next common, in frame, and then frame shift. Totally logical. This plot is the gene ontology summary. It gives us a look at the mutated genes and what they're doing functionally. So I see the words DNA binding and RNA binding in here, which is giving us a hint at what functional pathways might be impacted. The Circo summary plot shows where these variants are distributed in the genome, and they're pretty evenly distributed, except maybe chromosome 22, 19, 17. Those could be regions of interest. Could be random. The next tab of interest is the gene tab. So now we get into the real data. This table on the top shows each gene, 1 per row, and the table on the bottom, the section on the bottom, shows more specific information about that gene that wouldn't look very beautiful in a table. So here's what kinds of variants are impacted by that variant, and here's a visualization of the protein of the variants and where the variants lie. So if we do a quick sorty sort on this table, we can see that PD4-DIP has 32 variants, 20, 17, and 14 variants. We can very easily find which genes are the most impacted and even visualize where they all are. So see, for ataxin 1, they're very tightly clustered in this region. And weirdly, it's similar for AR, androgen receptor, 2 little clusters. Maybe I can infer that these are very conserved genes and proteins that can only tolerate variation in particular spots. And the different colors indicate different types of variation. We see a lot of blanks here, and that's largely because the way the data is sorted, but it's also because these particular genes do not have annotations. But if we wanted to look at genes with annotations, that could easily be added to the filter or sorted here. So every one of these genes is associated with some network in index congenital heart disease. And we can see that there are actually quite a few. But what's interesting is that for the most part, it's only one or 2 coding variants per gene that are already known to be associated with congenital heart disease. This could give us a clue that this is a very genetically heterogeneous phenotype, and maybe we're not going to find very many big hits where there are targets with lots of variants that we know cause the disease. Could be a constellation of variants, 2 to 3 per person. So those are some of the insights that we can get from the gene tab. Now the real fun, I saved the best for last, is the variant tab. It has all the annotations included that are variant level and a lot of the information from the VCF file. And I know 2,600 is quite a lot of variants, especially to not be included in the filter, but we can address that really quick with what I call local filters. Remember, we included segue heart to see variants that are transcribed. So now that we've taken a look at all variants that match the filter, we can further filter to view only variants that are transcribed in the fetal heart from Segway's predictions. That's still quite a few variants, but we can use other methods of filtering to track down known candidates. For example, which of these variants are known to be associated with phenotype in ClinVar? And some of these do actually carry consequences for heart, like Alström syndrome. But the thing I was most excited to show was the case control set. If we give this guy a click, and then, no, only just one click. It will sort the p-values from low to high. And I was very pleased as punch, to see that there are 3 variants that have a p-value less than 0.05, which is pretty good considering the kind of smaller sample size of the data. And these variants could genuinely be very impactful. Some insertions or deletions and a frame shift truncation could really affect the phenotype. These columns here show that this particular variant is seen 12 times in heterozygous state in patients with coarctation of the aorta, and only 5 times in patients without. Maybe this indicates, since sometimes coarctation of the aorta can be a subphenotype of the other phenotypes, like a minor feature, it could indicate that variants in CNO2, or maybe this particular variant in NO2, has some consequence. Sorry. Yes, CNO2 has some consequence for fetal development of the aorta. Another interesting way of looking at this data is to harvest the ClinGen gene annotations and find variants within genes that are known to be related to heart development and heart problems. There was one that I thought was really interesting, but it wasn't significant, so I need to take off that filter. And I think I need to take off the transcribed filter, which is why I didn't build this into the real filter, because sometimes these things don't play out exactly as you would expect. So some of these variants are quiescent, some of these variants lie within enhancers, and the interesting thing is they are all within a gene associated with familial thoracic aortic aneurysm and aortic dissection. So this is another way of finding variants that may be very plausibly related to the phenotype. And you can see that a lot of this more complicated information is displayed, like where the variant occurs in the phenotype, how many samples it's seen in, and we can overlay what the UniProt annotations are for functional residues in the neighborhood. So there's quite a lot of disulfide bonds happening in this region, but it doesn't look like the variant impacts one directly. So I hope with this example, at least, I've given a flavor of how this kind of modular tool can really be used to handle incredibly specialized analysis tasks, and maybe even given a couple avenues for collaboration, like tools or databases to add to the store. Thanks all for your attention and time.
So just 2 questions. I guess first, the more important one. This is super cool. Just curious about how one goes about installing the web interface and the backend associated with it for lab use or department-wide use. And then I had a technical question about just how VCF files are ingested. Is it lazy read off of a disk, or do you have to fully load it into memory on the web page?
It's not, okay. The first question about how it works in departmental, it can be put on a multi-user server and brought up in the GUI through that way with a single installation and a single version of the data. That way there's no need to duplicate it or like install it on your laptop the way that I have, although I find it pretty convenient personally. The second question about loading variants into the browser, that's something, that's the reason why I had to actually build in a filter. The graphical interface can't display physically more than 100 things at a time. Memory starts to bog down. So it is necessary to apply filters to load it below 100,000. But that makes sense to me because you can't physically go through that many lines in an Excel file anyway. So might as well do some filtering beforehand. And it can be pretty easily installed on servers or on a local machine with pip. So it's not really a particularly detailed install to be done.
Can I jam like variant files that I annotated via some other method into your viewer, or is there kind of stuff that the viewer needs that might not be there?
I don't think I have any examples to pull up, but I've done that with VEP. VEP can output an annotated VCF, and we can take in that VCF, and whatever information is in the, what's it called, the info field of the VCF will be displayed in in the viewer.
Great. Does it work with SNPF too? Because like the VEP and SNPF annotation, there's like a different tag, or is it just VEP?
So long as it's in some nice annotated VCF format, we should be able to load it in. And if not, just because we haven't tested with it with that output format yet, if not, just give us a holler on GitHub and we can adjust some things.
Awesome. Thank you so much.
Yeah, thanks a lot. My name is Minh Bui. I'm from Australian National University. So it's about 5 a.m. here, and I haven't slept so far. So in case my voice sounds a bit awkward, please apologize. So the project that I'm talking about is IQ3 software. And it's about our, recent progress on addressing several computational challenges in analyzing the SARS-CoV-2 data. And actually, I want to tell you that when we wrote the grant proposal last year, we never had this idea before. And actually, one of the aims in our proposal was to improve and speed up the IQ3 for ultra-large genomic data. And it just happened by chance that this type of data, indeed, a kind of ultra-large genomic data. So it's a very timely funding that we received from CZI to pursue this project. All right, so firstly, I want to acknowledge the team member who, without them, this project wouldn't be possible. So Rob Lanphier and me are the PIs on this project. James Babetti is a software engineer. He was recently recruited as part of the project. So he's directly funded by the CZI. And my joint postdoc with Rob, Thomas Wong, he's funded by another funding agency, but he also kind of entered and helped with some coding for this project. All right, so I firstly want to give you a rough overview, a very brief overview about phylogenetic inference so that people are on the same page. Nowadays, the data that we have is the whole genome sequence alignments, right? So that means you need to sequence the genomes and then align the genomes between different species like human, chimpanzee, mouse, and so on into a sequence alignment, right? So the step of going from raw sequence data to a sequence alignment is a prior step. And for us, we consider the alignment as the truth. So this data here can be seen as a matrix, where the columns of the matrix are the sites of the genome, and the rows correspond to the species. From this sequence alignment, we can use IQ3 or other tools to reconstruct the phylogenetic tree of the species. Yeah, so this is simple workflow, and I just want to note that before we started this project, actually the ultra large genomic data that we had in mind is are the data where you have very many sites in the alignment, but only maybe a couple of thousand sequence species, right? So that was the data we had in mind while proposing to pursue this project. And but nowadays, when we kind of early this year, when this COVID data kind of beginning of this pandemic, people started to sequence a lot of genomes, right, from many viruses, from many patient samples. And this figure is actually what I have shown in the CZI meeting in February. So this shows a phylogenetic tree of the coronavirus data back in February this year. And the tree is shown here, where the tips here are the samples. And you can see on the axis here, the dating of from the tree from December last year to February this year, right? And yeah, so this tree was built using a tool available at the nextstrain.org website. But in the background, Nextstrain used IQ3 to reconstruct this tree, right? And yeah, so one thing I want to focus here is you see that at that time in February, there were only a handful of genomes available, right? A little bit more than 100 genomes. So that was no problem for IQ3 at all. It can run very quickly on this data. So it can produce the tree within a few minutes. I was kind of following the literature and saw that people have been using IQ3 a lot, also for these data sets. So here are two papers published in Nature Microbiology and another one in Science, where the first paper is actually the paper where people kind of proposed to name the virus as SARS-CoV-2. And they have used IQ3 to reconstruct a small tree from this genomes data at that time from March. I believe, yeah, it's from February, March. And another Science paper where they try to develop vaccine for this virus. And in the paper, they also use IQ3. So these are just two representative papers that I found that use IQ3 for this kind of data set. Well, and I think many of you might know that people kind of are very much interested in this virus. So they started to collect the sequence and sequence the genomes very quickly. So as you can see, it grows from only a few hundred sequences in January very quickly, and up to today, we have nearly 250,000 genome sequence available. So this is a data repository called GIS8. So this is a website where they manage all the data. Right, so this now kind of propose a huge challenge to IQ3 and any other software, right? Not just IQ3. So here you can see that we have many sequences, and these sequences are very similar, and the sequences are rather short in the so-called phylogenomic era nowadays. So the genome is only about 30,000 base pairs long. But due to the many sequences, it poses a huge challenge for us, so for IQ3. So I remember that IQ3 could run the data set up to March /April, but beyond that, it could not run anymore. So therefore, James, when he kind of started the project back in June, his first task is to fine tune, so to speed up IQ3 for exactly this type of data. So he's a software engineer, so he's amazing in trying all different ways, software engineering techniques to speed up, fine tune the software in every aspects. So, for example, it might sound simple, but by kind of reorganize the memory access pattern, then he can certainly speed up some part of the code by 100-folds or more. And the speed up is actually increasing with larger data set, right? And he also employ vectorization techniques. So vectorization means that you can perform 2 or 4 operations, so addition or multiplication at the same time. And also try to employ multi-threading to further speed up the analysis, right? Yeah, so this is kind of a summary of his work, just one slide, but this work is for 2 or 3 months work here. And he had done some analysis with 17,000 sequence input data. So I guess 17,000 is around April. Yeah, that was data. So he could speed up the analysis. So it took the previous version of IQ3 took about 5 days to run on this data set. And after the fine tuning, it took only 8 hours. Well, it might sound still a lot, but that's kind of quite a very good achievement. All right, so Thomas actually tackled the problem from another aspect, meaning that he tried to parallelize the computation using distributed computing. So that means to distribute the computation across many different nodes in a cluster. And by kind of utilizing many different nodes available, then you can potentially speed up the analysis even further beyond a single machine, right? And there was one thing, so there's a library called MPI, or Message Passing Interface, that we are using here. And he spent quite some time to kind of design what is the best synchronization strategy between different nodes to obtain the best balance, best load balancing, and also to minimize the delay, so the waiting time. And something we learned from this project is that using asynchronous communication is actually not so good. So meaning that we found that for some reason with asynchronous communication, some messages kind of get lost in between, and we just don't know why. So therefore, we use synchronous communication. And we also found out that this latest version of the MPI library, they supported the one-sided communication. That I haven't learned before, but that's very cool. So here's some analysis that Thomas conducted actually just yesterday. So he tested from 1 to 8 nodes, and you can see that the speed up is linear when you have using 2 nodes. It's near linear if we have if we use 4 nodes. with 8 nodes, the speed up is only around 5. That means we still need to do something here to further improve the scalability of IQ3. All right, so Rob is actually the one who worked directly with the data. So Rob is actually working closely with the GIS8 people, where he kind of developed the pipeline that can automatically download the sequence from GIS8 and then process the sequence and build a tree from the process alignment. He has the data available in his GitHub repo here. So, and he kind of he's maintaining the tree on a very regular basis. There's only one thing that I need to convince him now, that right now IQ3 is actually not running. It's just too slow for more than, say, 30 or 40,000 sequences. So right now, Rob is actually using another software called FastTree to build this tree. But I hope that during the next months, we will be able to improve IQ3 to the point that we can convince Rob to switch to IQ3. Okay, so right. So as a brief conclusion, we kind of completed the first half, actually the second half of our proposal, where we want to improve the speed and the scalability of IQ3. The first aim, actually the first half of the proposal was about improving the modeling aspect of IQ3 so that the model would be more realistic for this huge data set. Actually, we also found this first aim kind of very well suited for this data, because one of the things that people had is sequencing error can be very severe for this type of data, because between two virus samples, there are only like about a handful of mutations. So a sequencing error can be very significant. Therefore, we have actually developed some sequencing error models, and we will test it on the data. And one future work in a very short term right now is, currently we construct a tree from scratch, right? That means even if we add new data to the existing data, we still build a tree from the beginning. But it would be better if there is a mechanism that allows you to keep the current existing tree and just add the new sequences into the tree. And if there is an algorithm like this, then it will potentially speed up the analysis a lot. And that can allow us to build tree for more than 100,000 sequences. All right, so with that, I would like to thank the funding from Chan Zuckerberg and other sources. Without them, it would be impossible to conduct this research and to maintain IQ3. And I'm happy to take any question.
So my name is Sarah Keating, and I work at the University College London. And the title of the project is actually Computational Biology Library Framework. And I'll sort of explain where that comes in a minute. About 25 years ago, computational power got to the point where people were actually able to do decent things with models on a computer. So people started to create computational models of biological systems. They also created software that allowed them to simulate or analyze those models, created results, and they published the results. However, to be truly beneficial to science, someone should be able to read a publication, recreate the model and interpret it in exactly the same way as the original author intended, run it in any number of different softwares that support a particular formatting of the model, produce identical results. But in order to do that, you need standards. And I have worked for many years in the area of standardization with the aim of encouraging modelers to create reproducible, reusable, not just models, but diagrams, simulations, and results. And as a result of the work of a number of people, there are now standards that will allow you to describe a model or to visualize a model, to describe the simulation experiment that you use to analyze the model, to illustrate just 3 of the standards that do now exist in this field. And about 11 years ago, people involved in these standards, many of whom sort of overlapped, came together to form the Computational Modeling in Biology Network, which is a community of people who are involved in various standardization efforts, who coordinate dissemination of information, coordinate meetings, coordinate producing guidelines on how to sort of organize the creation of a standard. So one of the main things we learned over the years is if you want people to adopt a standard, it's much easier to persuade them if you actually provide a software infrastructure that allows them to do it easily. Obviously, among other things, but that was my main interest. And I was involved in particularly the SBML standard, which is for describing models in the biological field. And we produced libSBML, which is an API for working with SBML. Originally, it was written in C++, but over the years has developed interfaces to a wide range of languages, which has always been at user request. And we still have languages on the request list, with Julia's the big one of the moment. So if people want a Julia interface. libSBML is completely open source, published under LGPL. And we did, until a couple of years ago, produce twice yearly releases where we created binaries that would work on all the major operating systems. And we averaged about 5,000 downloads per release. Also, as part of the work on libSBML, we created Deviser, which is a tool that would actually allow you to sort of propose an SBML extension and generate the relevant libSBML code, which was actually very useful in terms of developing extensions to a standard where you could sort of prototype very quickly and generate your code, go back and change it again, which was quite useful. But then, as all good things, the money came to an end, and we have had sort of 18 months with no input other than the community and people who are prepared to do something in their own time when they can. And it became apparent that more was needed to establish a code base that could be easily maintained. So now I am actually working for a research software development group, which is basically a permanent group within UCL so we're not specifically dependent on grants. We are paid to do our jobs, but to interact with different researchers throughout the university and providing sort of software development for them. So with that in mind, I applied to the CZI call with the main aim of providing seed funding to ensure long-term sustainability of libSBML and Deviser, with the goal of actually being able to expand this to other computational biology standards. And indeed to establish a base within a permanent RSC group, which would allow expertise to be shared and retained within the institute, so that, you know, when the money runs out again, we possibly have a better framework for letting it keep going. So to give you some idea of how old libSBML is, its original source control used was CVS. So it was then in SVN and has been in SourceForge for years. But if we want to sort of facilitate community contribution and whatever in the sort of this modern age, we need to move it to Git. So that was the first thing we did as part of the grant, was to pack it all up and move it to Git, which is not as straightforward as it sounds for an 18-year-old software that's already been through one change of version control. But we did manage to move most of the history. We did seem to lose a number of attributions, partly because people don't have GitHub accounts or we couldn't find them. But we got most of it's there, and most of the history is resolved or preserved. So that was the first sort of milestone for the grant, and we've already got that one done. And then the next aim is to incorporate continuous integration, particularly for the many language interfaces that we produce. Up until now, we did have local nightly builds, but that's obviously not ideal for people creating pull requests. And the other thing we want to do is automate the building of many of the language interfaces. And changing the CI and the builds that are which at the moment are currently spread around a variety of machines on the Caltech campus, to one centralized location should make it easier for members of the community to contribute and to access the binaries. And fortunately, we haven't actually gone down the Travis road because we're only just starting, so we don't have to address the issue with Travis becoming expensive. The next aim is to, having recognized that we could actually use Deviser to generate code for other XML-based languages, didn't just have to be SBML. But the disadvantage of doing that with the existing code is that the library you produce has complete dependency on the whole of libSBML, which is quite a large dependency, when all it really needs to do is depend on the bits that read and write the XML. So the second part of the aim of the grant is to actually separate libSBML into two libraries. Now, fortunately, the XML reading and writing layer of the library is already written to provide a seamless interface to whichever of the standard XML parsers a user chooses. So we are currently working on pulling this layer out of libSBML so that it becomes a library in its own right, which will be useful not just for people in the combined community, but for anybody using reading or writing, doing something that needs to read and write XML. The rationale for having this sort of layer in libSBML that is a seamless interface to the different parsers is that over the years we have discovered that researchers have very fixed opinions about which XML parser they wish to use, and so it was just easier to allow them to configure whichever one they wanted, and just make it seamless for the interface to use. So having separated the libraries, that will actually make it a lot easier to use Deviser to generate APIs for other XML-based standards, because they will need to only the dependency on the XML reading layer, and not the rest of the SBML stuff. So hopefully we can use Deviser to generate APIs for things like SBGN and ZML and other languages that exist within the combined community. And sort of that, which leads on to leads on to our third aim, which is basically update Deviser to generate this code for other XML-based standards. which in the course of doing that, we desperately need to create some developer documentation for Deviser. It has some very nice documentation for how to use it to create a level 3 SBML level 3 package, but it has nothing that really explains what's going on under the hood. So obviously, to extend the expertise and spread it a little more widely, so it's easier for people to contribute, we need some good documentation. So the overall hope for the grant is that it will facilitate future community involvement in both libSBML and other libraries in the combined network. And we're hoping that by establishing a permanent RSE base for API maintenance, we can encourage people to maybe contribute even tiny amounts of money from their grants where they use our software to actually allow us to actually have at least one person who is paid to maintain or at least oversee maintenance, and hopefully keep the libraries up to date for the foreseeable future. And with that, I acknowledge the guys in the group who are also working on this, so they're the ones who I'm disseminating the knowledge and information to. And Frank Bergman and Brett Olivier, who are both members of the combined community who are acting as collaborators or as consultants in their free time. And I would also like to thank the Chan Zuckerberg Initiative for recognizing that open source software for science is indeed essential. Thank you.
Yeah, quick question for Sarah. You mentioned spending a lot of effort on getting the history moved over. I was curious, you know, an 18-year-old project, some of the early commit history probably isn't all that relevant. Did you feel like with an open source piece of software that there's some obligation to try and preserve, you know, who it was that
that
was part
was
of the legacy?
The main incentive was that it felt very unfair to sort of put the library into GitHub and say that it only had 2 contributors, when in fact it's had sort of 20 or 30 over the years. And it just seemed sort of counterintuitive to the open source sort of ethos.
Yeah, I mean it's interesting because unless they have a GitHub ID, it's very hard to put it in there. So I wonder if there's some sort of suggestion back to GitHub on that as a legacy item of some kind that they could allow us to do.
Yeah,
but anyway, interesting. Yeah.